Thanks everyone for attending this next instalment of the Arthur Rylah Institute seminars. For people who might not know me, my name's James Todd. I'm the Acting Chief Biodiversity Officer and Director at Arthur Rylah Institute. So it's my pleasure to welcome everyone here today and to chair the meeting. I've also got members of the team here, particularly Tess and Penny, who are on hand, who will be sharing links and monitoring the chat for questions. So we hope to be able to compile the kind of some of those questions for at the end of each of the presentations. But before I start, I'd like to begin by acknowledging the traditional owners of the land which we're variously coming from, and people might be coming in from all parts of Australia. I'm on the land of the Wurundjeri people here close to Melbourne, and pay my respects to the elders past and present, and also to any traditional owners and or Aboriginal people who might be joining us on the line today. We know that invasive animals are a major impact and risk and threat to biodiversity conservation. Our ability to mitigate the impact of invasives is highly dependent on robust science that can help us make decisions, cost-effective decisions about control and potential eradication. As a demonstration of that critical science, we've got two fantastic presentations here today from ARI scientists in Lauren White and Justin Kelly, and they'll both in slightly different areas and using different approaches, but they'll be highlighting some recent research that's come out of ARI and how it's being used to inform decision making and potentially, how it might be used in the future. To start us off, Dr. Lauren White is talking about her investigations into feral pig population dynamics using DNA analyses. That will follow by Justin Kelly from also from ARI, who will give us a presentation on recent statewide deer survey and modelling that's providing us with a comprehensive understanding of the distribution and population of deer on public land right across Victoria. As I said, we'll have time for questions after each talk, so if you've got some questions, please put them in the chat, and Tess and Penny will organise those so that we try and get to the ones which are perhaps the most popular, and noting that we won't probably get to all those questions, but Justin and Lauren might be able to respond to those after the session. So without further ado, a couple of presentations that I'm really looking forward to. I'm going to hand over to Lauren. Thanks, Lauren. Away you go.
Thanks, James. All right. Hi, everyone, and thank you for tuning in to hear about our study on feral pig population genomics. So pigs arrived in Australia with the first Europeans, and wild populations started to establish pretty soon after colonisation began. Obviously, we only have patchy information about the distribution early on. This is the first map I could find of pig distribution. It's from 1947. So you can see they are present in patches of WA, Northern Territory, most of Queensland, northern New South Wales, and then this riverine region in southern New South Wales. And they've considerably expanded their distribution since then. This is pigs in 1982. You can see they've now started to reach the northern parts of Victoria. A more detailed map from 2008 shows that they are now distributed across much of eastern Victoria. And this is the latest map I could find from Feral Scan, where the public can record sightings of feral pigs. Red indicates areas of highest density of sightings. And you can see that the southwest of Victoria and central Victoria are some of the most recently invaded regions. And this distribution continues to expand, which is bad news because feral pigs are one of the most damaging invasive animals globally. In Australia, they'll prey upon and compete with livestock and native animals. They consume and trample crops and native plants. They cause soil erosion, water degradation, and they pose a very significant biosecurity risk. They can act as vectors to some very nasty exotic diseases, and so their presence on the landscape is a threat to both human health and agriculture. For this reason, many organisations and governments undertake programs to manage feral pigs. This can include baiting, trapping, fencing, and often aerial or ground shooting. As you can imagine, these are time-consuming and expensive undertakings, but they can be effective tools in managing feral pigs when implemented with care. However, our ability to optimize control program design is limited by our lack of understanding around feral pig movements and dispersal characteristics, which is where our study comes in. We aim to use genomic data to increase our understanding of feral pig population connectivity and dispersal and movement patterns. We hoped to be able to inform control programs and management. For example, we hoped to be able to identify isolated populations that might be good candidates for eradication. And we expected to find at least some regions where connectivity was very high, implying that control would need to target large areas to prevent rapid reincursion. And we also wanted to improve our understanding about how feral pigs were moving into and colonizing new areas. This information could be used to improve monitoring, early detection, and containment. So to do so, we worked with state agencies and their contractors who collected biopsies from culled pigs during control programs from 2017 to 2021. We processed 607 samples, but for most of the analyses I'm going to be presenting today, we're working with a downsampled data set where we excluded juveniles and made sure we had even sampling across the 20 regions that we collected from to prevent bias. We genotyped samples using Illumina bead chip technology, which allowed us to generate data for 39,000 markers after filtering. So it's a very large data set. And we first used this data to examine genetic diversity. Here I'm presenting heterozygosity. It's a common measure of genetic diversity, summarized in this case by sampling region. If you look at the map, you might notice a couple of pockets of high diversity that look familiar. These reflect the regions from which feral pigs have expanded since the 1950s. That's the Riverine region down here, and northern New South Wales and southern Queensland. So this is a common pattern with invasive species. When they're colonizing new areas, it usually involves a population bottleneck, which decreases diversity. And you can see some of the lowest diversity regions are the newly invaded parts of central and southwest Victoria. So basically diversity is highlighting their invasion pathway, which is nice. It confirms our understanding of past pig movements, but we next wanted to tackle some questions that might help control programs in the immediate future. So we moved on to clustering. Now, when two populations are isolated or gene flow between them is low, they're going to accumulate genetic differences over time. So techniques that cluster individuals based on genetic similarity can highlight populations that are isolated. We used a few different approaches to do this. I'm just going to present one. This is a PCA plot. Each point is a sampled individual, and their placement in relation to other samples is indicative of genetic similarity. So you can notice there is some geographic patterns here. Individuals generally cluster or group rather with individuals from their sampling region. And we've got three broad, I'm going to call them branches for want of a better word, that reflect some spatial patterns. So this branch represents individuals from this region, we've got a northern group, and then this sort of widely distributed but still geographically contiguous region. However, you can see that the way I've defined these branches is very arbitrary. You could easily have drawn this slightly differently depending on the way you squint at the patterns. And this is because there's no strong delineation between any groups on this plot. You'd be really hard pressed to confidently draw a line and group them separately. And these results suggest, and our other clustering analyses confirmed, that our data doesn't fit clustering models well. It indicates that we don't have strongly isolated populations in this data set, and in fact suggests that we've got a high level of connectivity, at least between neighbouring populations. So where clustering is trying to define groups based on genetic similarities, clients describe a continuous pattern of genetic diversity in space. Because dispersal is always limited, there's only so far an animal can move in its lifetime, genetic differences between two individuals usually increase with the geographic distance between them, which creates a cline. So this plot, we've got each point is a pair of feral pigs. The x-axis represents the geographic distance between where they were sampled in kilometres, and the y-axis is a measure of genetic distance or genetic dissimilarity between two individuals. The blue line is the correlation between genetic and geographic distance, and as expected, there's a positive relationship, but it's pretty weak. That line is nearly flat. But if we zoom in on this part of the plot, just looking at pairs that are separated by up to 250 kilometres, it looks like something more dramatic's going on. And indeed, yes, the correlation between genetic and geographic distance is stronger here. We can zoom in again and we find that it's a bit stronger again. This is a common pattern in genetic declines. There's a strong positive relationship between genetic and geographic distance at the shorter distance classes, but that relationship decays and eventually plateaus to the point where there's no relationship when two individuals are further away. And the really neat thing is this rate of change and the point at which that relationship turns to zero contains information about dispersal patterns. And we can analyze these plots to draw out that information. Now, without going into too much detail, because I don't have the time, what we find is that the correlation between genetic and geographic distance is strongest at the very shortest distance classes, 5 kilometers or less. And the decay in that relationship is really quite rapid. It nears zero at about 50 kilometers, but remains weakly positive out to around 100. So what we take away from this is that most pigs are not moving very far from where they're born, 5 kilometers or less. However, longer distance dispersal is occurring up to around 100 kilometers, but those longer distance dispersals, 50 to 100 Ks, are probably quite rare. So we can confirm these dispersal patterns by examining something maybe a bit more tangible by looking at close kin. We can use our genetic data to identify relatives in our sample set. And firstly, we're going to look at first degree relatives, that's parent offspring or full sibling pairs. And the distance between first degree relatives can act as a proxy for natal dispersal distance. That's the distance between where an individual was born and where they eventually breed, or in our case, where they were sampled. So we found almost 2,000 pairs of first degree relatives in our sample set, the majority of which were sampled very close together. The average distance between adult first degree relatives, that's important, we're only looking at adults here, the average distance was 4.8 kilometers. Again, highlighting how pigs are quite sedentary in nature and showing that most dispersal events are short or negligible. We do find some long distance events, including a pair of males from the Snowy River region that were sampled almost 60 kilometers apart. Again, showing that long distance dispersal is occurring, just rarely. We can take this a step further. We can look at second degree relatives, which includes grandparents and grandchildren pairs. This is Peppa Pig and her grandparents, for those who don't know. It also includes half siblings and aunt, uncle, niece, nephew pairs. So these aren't a proxy for natal dispersal like first degree relatives are, because these relationships can include more than a single generation, usually 2 or 3, but they still provide information about movements. The average distance between second degree relatives was 32 kilometers, which is reasonable. It's within the range of what we'd expect given everything else we've learnt. But the maximum distances between second degree relatives were a surprise to me. So I'm going to show you on this map. These points are our sampling locations, and I'm going to draw a line between the second degree relatives that were separated by the most extreme distances. So there's two sort of areas to focus in on. We've got individuals from the southwest of Vic, there's 3 of them, that have second degree relatives more than 800 kilometres to the north in the northern parts of New South Wales. Also, the Otways has a number of individuals with connections more than 400 kilometres away in southern New South Wales. So these extreme distances, we believe, are in no way achievable through natural pig dispersal, even over a couple or 3 generations. It's a literal when pigs fly scenario. Given this and the fact that these patterns are quite localized, we take this as evidence of illegal translocations, where pigs are being trapped from one region and deliberately released in another. We are assuming that the direction of this movement is from the more established populations in New South Wales into Victoria to deliberately expand their distribution and augment hunting opportunities. So while this was a surprise to me, it became clear after talking to our partners and collaborators that it's not a surprise to those living and working in the regions. There's an awareness that this is occurring in Victoria and South Australia where pigs are being released. This is an ABC News headline from the end of last year. And there's also an awareness from our partners in New South Wales where pigs are being trapped and moved from. So what we found is a confirmation, an extra line of evidence that these illegal activities are occurring. All right, so there's a final set of analyses I'm going to present. so far, everything we've presented is looking at a very broad scale, but control usually occurs regionally, and so analyses at local scales might be of use. So for today, I'm going to zoom in on the southwest of Victoria region. These points all represent sampled individuals. There's 125 in all. They're sampled from 8 different sites, which were mostly commercial plantations. But we've also got 1 private farm and Budge Bim Indigenous Protected Area. So since we were just talking about illegal translocations, I'll firstly point out that the 3 individuals with close kin in the distant New South Wales population, they were sampled here where the white triangles are. They're all sampled in commercial plantations, which hints at really that certain land management practices might have characteristics which make them attractive as release sites. So for example, plantations might have a larger number of unmonitored access roads. So that sort of information might be useful to improve monitoring and early detection. However, these are only 3 data points, and we don't think these individuals specifically were translocated. It might be their grandparents or parents, because all 3 have a number of relatives within the southwest of Victoria region. So I'm going to put these guys aside for a moment, and the rest of the analyses we're looking at for these local analyses, we're going to assume that the patterns are driven by natural dispersal, and I think there's good reason for that assumption. So as I said, we found a large number of relatives in this region, mostly within sampling sites, but also some between them. So these lines represent first-degree relatives. You can see there are 2 main family groups, one in the east and one in the west. And these family groupings drive the clustering that we find, as you can see on the PCA. The nice thing about this region is we've got a number of dispersal events for which we have direct genetic evidence. So not only do we have all these first-degree relatives connections across sites, which are, as we said, proxies for natal dispersal, you can also see on the PCA, we've got this one purple individual. So he was sampled at the purple site, but he clusters with the yellow group. So all his family are up here. So that's a direct evidence for a dispersal event. We've also got this pink individual that falls intermediate on the PCA between the 2 groups. This suggests that he has 1 parent from the west and 1 from the east, showing that there has been dispersal between these family groups also occurring. So taken all together, we're again confirming that pigs are not moving very far from where they're born. But this local analyses highlight that this is probably for social reasons. They're staying within family groups. We also show that despite their mostly sedentary nature, neighbouring families are connected via dispersal events over short to moderate distances. So just to give you the scale, the distance between the purple and the yellow group is about 20 kilometres. And this all links back to what we found in our clustering analyses right at the beginning. Despite short average dispersal distances, adjacent family groups will be connected. And where you've got a contiguous region of family groupings, it's going to translate into a highly connected metapopulation across a large area. Okay. So I realized that was a lot. I'm just going to summarize the main takeaways from this study and then link them back to how I think this information can help control programs. So firstly, we find that feral pigs are relatively sedentary and they're often remaining with their family group without dispersing far. That's not to say that dispersal is not occurring. Our results show how neighbouring family groups are connected via dispersal occurring at short to moderate distances, and that pigs are capable of dispersing up to 100 kilometres. At a broad scale, where we have a contiguous distribution of feral pig groups, this translates into a highly connected metapopulation where we don't find any evidence of strongly isolated populations. And finally, we've found evidence of illegal translocations, we think, into sites where pigs are being released into sites in Victoria. So what does this mean for management and control programs? Well, a high level of connectivity we found shows that reincursion risk after control is high across the study area. This suggests that control efforts may need to be ongoing or target large areas to be effective in the long term. High connectivity also has implications for biosecurity. If a disease outbreak does occur, our results suggest that it could spread through the feral pig population rapidly. It's not all bad news though. Since we found that average dispersal distances were quite short, we think there is scope for coordinated control efforts to target regions in a way that could break up this metapopulation, isolate some regions, and reduce biosecurity risk. This would probably require more research, definitely careful planning and cooperation across regions, but it could have substantial benefits. However, illegal translocations are clearly a problem, and they're very likely hindering control program effectiveness. It was very disappointing to see that the regions where we think are being targeted for release of pigs moved from elsewhere were areas that until recently, they didn't have established pig populations. So this suggests that illegal movements of pigs is contributing substantially to the expanding distribution of feral pigs in Australia. I don't have solutions to this problem, and I can only point it out, but I'd suggest it requires a broad range of stakeholders coming together to address this issue. All right, thank you for listening. Thank you to Agriculture Victoria for funding this research. Thank you to my co-authors who are listed down the bottom there, and our partner organisations who helped us with the big job of collecting the samples for the study. I'm happy to take questions if there are any.
Thanks, Lauren. That's great. Lots of rounds of applause there for you in coming up on the screen. I think we also know or suspect that hunters are illegally transporting deer around as well. So the fact that you're finding it in pigs is perhaps not not as surprising. You mentioned obviously feral pigs are a potential disease risk, and that there's some of that risk of getting into say Victorian piggeries. Have you a sense of how this research might could be used to inform what those risks are and potential options to kind of mitigate that risk?
Yeah, so we haven't done it for this study, but the potential's there to estimate migration rates between populations. So at the moment, we've been looking sort of very broad scale and average distance dispersal distances, but there is definitely scope for doing more fine scale analyses where we look at landscape features or control decisions that are reducing migration rates, which is that's a very important aspect of the rate at which a disease would spread, is how fast, how connected are two populations. And so for sure that there's scope to use this sort of data to answer those sort of questions for both biosecurity contexts.
Right. So we've got time for a few answers to some questions. So there's a question there. The first one popped up from Craig was around the sampling and whether animals were destroyed or whether you release some with radio tracking collars, such as Judas pigs that are used in trapping programs. I understand these are actually from destroyed animals, aren't they? The sampling.
Yeah, yes, they're all from culled pigs. I am aware of some of those studies that are ongoing, but as far as I know, all of our samples came from destroyed pigs. Yeah.
Yep. And is genetic work continuing in any shape or form?
We're still collecting, so we're still collecting and archiving samples. We don't have plans for the immediate future for doing any more genotyping, but we're certainly collecting, and if there's interest, the ability's there.
Great. And there's one there which is, I don't know, can you actually see the chat?
I can see some.
Okay.
Yeah,
yeah, I'll let you just have a look at. There's one there about whether the genetic clusters indicate, whether the pigs might be suitable for eating or not.
Oh, that's an interesting question. Yeah.
Well, I guess also because there's, we know that there's lots of disease in feral pig populations.
So the genetic data that I generated can't tell us whether or not a pig is diseased. We could possibly compare it to farmed breeds to see which are more like farm breeds, but no, I don't think we could use the data I specifically generated to answer those questions. The DNA is there, the samples are there, so it's possible you could do further testing from those, but not with the data I generated, I don't think.
No worries. Maybe just time for a few more. So there's a question there about reincursion risk. You said reincursion risk is high.
Yeah.
Can you elaborate a bit more on the basis of that? How you came to that conclusion?
Yeah, I guess it's a relative term. mostly we're looking at if two populations are connected with gene flow, if you have control this area, it's likely that animals are just going to move back in from next door. So I haven't quantified that, but like we were talking about earlier about the estimating migration rates, that's possible. We could do something like that where there's interest in specific regions. Yeah.
And notwithstanding pigs being moved around illegally.
Oh, yes, exactly. That complicates things.
Those isolated, perhaps those more isolated populations that are, perhaps greater than 5K distance from others, there might be some greater likelihood of eradication if we're able to bring that necessary control effort to those. Yeah,
so you could use those average dispersal distances to sort of to pick ones that look like they're more isolated.
There's one there about whether you've, the issue that you found with the alleged illegal transporting into plantations. Has that been raised with any of the plantation companies in the southwest that you're aware of?
Not that I'm aware of. It was Parks Vic who helped us collect those samples in collaboration with the plantations. I haven't had a conversation with anyone from there. No, not yet.
Right. Okay. Maybe the last one then, before we move to Justin, is this research being used to target localized removal efforts and where it would be most effective?
Not at the moment, although there's ongoing discussions, but no, not yet. Very early days, I think.
Great. All right. And there's a question there about diseases preclude eating. I think we might leave that one for. There'd be plenty of online information about what diseases feral pigs carry. Okay, thanks, Lauren. That was fantastic. Really informative and some really interesting research results there, and hopefully we can apply some of that more in the future, both in terms of benefits to agriculture, but also to the natural environment in terms of feral pig control. Okay, so next we're moving on to Justin Kelly. So Justin's going to present on his recent research into the abundance and distribution of deer on public land in Victoria. Over to you, Justin. Thank you.
Yeah, thanks, James. So today I'll be talking about some recent research that ARI has done, primarily myself and Dave Ramsey here at ARI, and we essentially looked at the abundance as well as the impacts of deer on Victorian public land. So by deer, I mean in Victoria, the 4 established species of deer. This includes sambar, fallow, red, and hog deer. So before this study started in 2021, we really didn't have good information in Victoria on a couple of key important knowledge points. We didn't know how many deer there were in Victoria. We didn't know the range of these deer species, so we didn't have really good or precise and up-to-date measures of the range of deer, so where they occupy in Victoria. And we also didn't have an understanding of their trends in abundance and distribution, more of the short term. We can compare against, say, 1980s or early 1900s, but in terms of the trends year to year or every 5 years, we didn't really have that information. So without that information, it's actually very difficult to effectively manage deer in Victoria and also evaluate the management actions to make sure those management actions are having a benefit or the desired effect. So that's where we come in, and that's where our population surveys come in. So essentially, by conducting population surveys, we're able to answer questions of how many deer there are, where they are in Victoria, and we're able to form essentially the baseline to measure trends moving forward in the abundance and distribution of deer. So how did we go about monitoring deer population in Victoria? Well, we measured quite a few things. So at a site, one of the first things we did was to measure the vegetation at a site. So this will give us an estimation of what sort of impact deer are having. So the vegetation we measured included a range of different structural vegetation components. This included seedling counts, sapling counts, herbaceous understory, native woody understory, as well as the presence of exotic species or weeds. We also deployed camera traps, which I'll talk a bit more about later. And these were sort of the crux of our monitoring. And we also walked deer signs. So walking deer, 3 different transects looking for different deer sign. And by deer sign, I mean deer pellets, deer footprints, deer rubbings, which the males make during the breeding season, and deer wallows. So with regard to the camera trap, what we did was we deployed a camera trap as well as distance markers set out here at 2.5, 5, 7.5, and 10 meters. Essentially what this does, it enables us to not just measure what species of deer comes in front of the camera, but at what distance they come in front of the camera. Essentially, through some statistical models, we can use this information to calculate the density or abundance at a particular location. And therefore, we can extrapolate that for the density at a particular region and then a density or abundance across the whole state. Essentially, this is richer information than just presence-absence data, and that allows us to calculate abundance of deer across the whole state, or across all public land in Victoria. So where did we conduct this monitoring? Well, over the space of 2 years, we deployed cameras and had our sites at 317 locations. This was all on public land in Victoria, but it ranges from Far East Gippsland up to the northwest of Victoria. It included close to 17,000 camera trap nights, which produced well over 1 million camera trap photos, and hundreds of kilometres of transects were walked. All the deployments were done in the months of summer, spring, autumn. So just keep that in mind, that in winter there might be some differences in deer distribution, especially in the Alps, that we don't capture. So using these field methods and these surveys, how did we put that together to really estimate deer abundance? Well, I'll just walk through now the different components we used to, I guess, combine together, mush up together to estimate deer abundance. So the different components that we fed into our model were, of course, the camera count, so how many deer came in front of the camera. We also took into account the various environmental variables at a site that might dictate how many deer might be present at a certain site, and that included environmental variables such as the bare soil composition, the nitrogen composition, things like distance to pastoral land, knowing that that's a good resource often for deer to use to browse on, as well as other climatic components such as precipitation and the amount of forest edge surrounding that site. We also took into account a random effect of bioregion, and this is really an effect that sort of segments different environments in Victoria that might have underlying reasons why they are better or worse to support different deer species. We, of course, needed to take into account detection probability in front of the camera. And briefly, this is the idea that when a deer walks in front of the camera, it's not guaranteed that that camera will trigger and you'll detect that deer. So essentially, our distance sampling enables us to account for imperfect detection of deer, which is really important to estimate the abundance or the true abundance at a site. We also need to account for activity, knowing that deer are not active for a 24-hour period of time. So they're actually only active or estimated to be active about half that time, of 12 hours. So we need to take that into account when we're modelling abundance. We also need to take into account group size, as this will affect both the detection and the abundance of deer if there's more deer coming in front of the camera. And lastly, we of course need to take into account effort, so how long the camera was deployed for, because the longer you deploy a camera for, the more likely it is you'll get more photos of deer. So essentially using this model, that we can use that and we can distill that down into getting an estimate of deer density. So deer density would be a set amount of deer per square kilometers, and that would be specific to a site which has a set of environmental variables which we measure and we know for the rest of Victoria. And because we know that for the rest of Victoria, such as things like the soil nitrogen or the bioregion, we can essentially predict deer density to the rest of Victoria and come up with then estimates for density across Victoria and total abundance in the different regions on public land. So now moving on to the results. So essentially what deer densities and abundance look like on Victorian public land. So firstly, let's have a look at sambar deer. We estimated an abundance of about 123,000 sambar deer on public land, with that range going from about 96,000 to 157,000. With key hotspots, you can see here in the northeast, around that Omeo and Dinner Plain, as well as in the East Gippsland and closer to Melbourne. Secondly, we see fallow deer has a much more patchy distribution, but their estimated abundance is about 50,000, with that range going from about 30 to 85,000. For fallow, importantly, key hotspots are in the southwest, around Budgebim and surrounding areas, but there's also a patchy distribution, of course, in the northeast, as well as parts in the Otways, and really a lot of places in Victoria. For red deer, we estimated a smaller abundance than the other two deer species, with about 12,500, and that range there is quite large and broad, so a bit more uncertainty with this deer species. But we can see that the key hotspots are over in the Grampians as well as in the Otways. And lastly, looking at hog deer, which have a much more confined distribution to the South Gippsland, we estimated the abundance of this species at a bit over 4,000 deer, with that confidence interval ranging from about 2,000 to 8,500. And you can see the key hotspots here are Wilson's Prom and then coastal areas of South Gippsland. Okay, so that's our abundance or density distributions across Victorian public land for the different deer species. So if we combine that together for total deer on public land, we get an estimate of about 190,000 deer on public land, with that range ranging from about 150 to 250,000. So the next question is, because it's often a bit hard to really interpret and understand large numbers, is what do these deer densities look like, and how does it compare with other studies? So if we have a look at our deer densities across the range, we see that on average, these for the different deer species, we get deer density ranges from about 1.5 up above 3 deer per square kilometre for the different deer species. You can see here with species like sambar being the largest, as it occupies a real large chunk of Victoria in the east. Now, to compare this against other studies, we actually looked at 33 other reported studies that estimated deer density in Victoria and Australia. These were often a lot smaller scale studies, so in a particular reserve or catchment or national park. And from these studies, we found that the mean estimate for density of deer was 4.1, and the median was about 1.7. So essentially all our deer estimates here for our different species are in the ballpark of those existing estimates and what is known and expected for deer population size or deer density in Victoria and Australia. To highlight this a bit more, I'd compare it against one other study that was in Tasmania. They also used distance sampling, but they used distance sampling from a helicopter here. And flying transects in northeast Tasmania, they were able to estimate the density of fallow deer, the fallow deer population. And they found in an area about 20,000 square kilometres, an average abundance of about 53,000 fallow deer, which corresponds to a density of about 2.7 deer per square kilometre. So again, quite similar to the densities that we produced in our report. So this might be, for some people who are familiar with deer in Victoria, you may have previously heard a suggestion that there might be 1 million deer in Victoria, and it is sometimes reported as such in news articles and the like. So just to hit a few discussion points without going too deep into it. Firstly, get that first point up. So we only estimated deer abundance on public land, which is 75,000 square kilometres. Now, there's about another 25,000 square kilometres of private land with tree cover that can support deer populations in Victoria. So there's a chunk, another quarter, I guess, of potential habitat for deer outside the public land that we monitored that would be holding extra deer. But even then, that 1 million number seems quite high and quite difficult to get to based on the deer densities that we found. So we're actually unsure of the original study suggesting that there's 1 million deer in Victoria. If it's related to the hunter surveys, keeping in mind that hunter harvest estimates of 100,000 per year are reliant upon both the accurate and truthful recollection in those surveys of how many deer were harvested by hunters in each month across those reporting periods. So if there was 1 million deer in Victoria, density would have to be much higher than what we found and what other studies have found. So that it would really have to be an outlier. And lastly, 1 million is a round number. So there is such a thing as a round number bias, and we tend to like using round numbers to report things, I suppose. So moving on from abundance now to the impact of deer on vegetation. So we found deer density affected various structural vegetation components. Firstly, deer density, so an increase in deer density at a site, led to a decrease in native woody understory. So these are usually the shrubby layers around a site. Conversely, it led to or was related to an increase or higher level of native herbaceous understory, so lots of grasses. And an increase in deer density also was related to a higher presence or likelihood that exotic flora species would be present. So where to from here? Well, firstly, one aspect we could do to improve our understanding of deer in Victoria is to conduct more surveys on private land, perhaps through use of aerial surveys. Secondly, we would really like to see those spatial data sets that we've produced in this research be used to inform deer management. And I think Penelope will post those in the chat, those links to access that data from DECA's data share website. Thirdly, what would be really great is to have repeat monitoring of the sites that we measured here, and that would be for use in various biodiversity indicator frameworks, such as the VBX or Victorian Biodiversity Index, which can help us track how we're going over time in terms of managing the impacts of feral species such as deer. And lastly, what's great about camera traps is that they collect a lot more data than what you're usually targeting. So here's just a list of the different species that were recorded at our 317 sites. So essentially using this data, we could produce abundance models for a lot of different species in Victoria, whether that's macropods, in wallabies and kangaroos, or whether it's even emus, or one recently, dingoes, which are very topical at the moment. So in summary, to draw a few conclusions, we estimated total deer abundance at about 190,000, with that range from about 150 to 250,000 on public land in Victoria. So that's 75,000 square kilometres. So density estimates that we found largely align with those previously published. Increase in deer density at a site related to lower native woody understory, higher herbaceous understory, and higher likelihood that exotic flora species would be present. We can only really get trends in abundance over time by repeat monitoring. So to really tick that box, we've got the baseline now. To move forward and really track that over time, we would need to do repeat monitoring. I'd just like to close by thanking a lot of people who've contributed to this project from ARI, but also externally across DECA and different organisations. Yeah, if you're interested in understanding this more, we've got our report that's available from the ARI website, which you can look at into more. And yeah, I think that's a wrap from me. So yeah, thanks for listening.
That's brilliant, Justin. Thank you. Lots of rounds of applause there. We've got some time for some questions. I might just ask a couple of lead-in ones. So this research has been funded out of the Victorian Deer Control program, which sits under the Deer Control strategy. How do you think, or what's the best way that this information might be used to inform management?
Yeah, I think there's a few ways it can inform management. Firstly, I think it can be really great in terms of providing context for control. So having an understanding of, well, what's the existing estimate of how many deer I have in my particular region or in this particular national park, and then put that in terms of the context of, well, if I'm doing some sort of control, how many deer have I taken out from this? Am I making a significant dent in the estimated population size or not? So that context is quite important for it. It can also be very useful in terms of knowing where might be the hotspots to control deer at that broader scale. So, you know, are there particular national parks or regions that might have or appear to have higher deer density that we can focus on? And how does that really overlay with maybe your strategic values in terms of biodiversity on the ground? So again, that data can be sort of overlaid in terms of what are my underlying assets, and then how does that really look in terms of, yeah, where I should focus control on. So that's a couple of different ways, yeah, it can be used for management.
And you mentioned the literally millions of images that have been captured of various species, and you referred to some recent work that's being done in terms of estimating the population of dingoes across Victoria. Do you want to talk a little bit more about how what you've done to kind of supplement the or use of the original 317 cameras across Vic and what you've done to kind of supplement that to get a handle on dingo population.
Yeah, Dave and myself have managed to almost produce a similar estimate for dingoes in Victoria, again using the camera trap data. And again, because when a deer walks in front of a camera, the camera gets triggered, and when a dingo walks in front of the camera gets triggered. So essentially, we have that data collected. And so it's about essentially rerunning the model, of course, with different components of what might explain dingo density in Victoria, as well as, different likelihood of detection. And that was supplemented with additional surveys that were done in the Northwest as well, because we didn't extensively survey the Northwest in this deer project. As you might see from our surveys back here, where they were. Get it up there. Yeah, we didn't have heaps of surveys in the Northwest because deer are not largely present throughout a lot of those areas. So yeah, supplementing with additional surveys.
That's great, thank you. All right, so we've got some questions in the chat. There's a question there about can you draw any conclusions on how landscape scale fire is driving deer abundance and overall populations in Victoria? For example, it's suggestions that dense fire regeneration regrowth forest promotes deer abundance.
Yeah, it's a good question, and there's some existing studies that have looked at the response to fire and how that impacts deer abundance. It's quite hard for us to conclusively state the impact of fire because we don't essentially have a before and after measure of abundance for, say, the 2019/2020 bushfires. A lot of our surveys were done, all of our surveys were done after those big fires that affected large chunks of East Gippsland, which are a Sambar stronghold. Possibly one interesting thing is that, maybe some of the densities, as you can see here in parts of East Gippsland, were a bit lower than maybe we expected, or some other areas in the high plains. And that might be due to the impacts of that fire initially taking out probably large amounts of deer. However, they do colonize those areas. So again, this is where the repeat monitoring would be very interesting, because potentially now, as it's already been 4 years after, or in the next couple of years, we might see the density in those areas in East Gippsland to be quite a bit higher than what we originally estimated with. But also that depends. There was a lot of active aerial control going on in those areas over time. So it's not like a, I guess, natural experiment where they're just recolonizing without interference. There is management to consider on top of that.
Thanks, Justin. So maybe there's a few questions relating to management, and obviously DECA did stand up a large aerial control program straight after the fires back in 2019-20. So a few questions. Could some of the animals that have been controlled through those programs be used to add to the density data set? And also, could this data set be used to assess the effectiveness of some of those management interventions?
Yeah, for that aerial control data, Dave's done a study and published a study, which hopefully will get put in the chat, maybe on estimating deer density from catch effort models from aerial culling that took place there. And that study was one we compared our estimate to, and again, we found that our statewide estimates for that region, for those national parks in question that had aerial control, were very similar to the density estimates that were made from that aerial control program. So really, it's a quite useful study in terms of comparing those density estimates to completely different methods used to estimate density, and essentially seeing if they align. So yeah, that was quite useful.
And then the other question was around could the kind of camera trap data be supplemented by data that might come out of various deer control programs?
It can be. It depends on how you combine that data and how you statistically model it. So again, in our study, we combined camera trap data as well as transect data at a site because there's a probability, for instance, that you don't detect a deer on a camera, but you see it on a transect. So you know that there's deer there, but it's it's unlikely that deer are there at a high enough density that you're seeing them on the camera a lot. So there's ways to incorporate all this different sources of data into these estimations, but a lot of the time, if you're using multiple methods, you need to, again, think about how you combine them together, and that requires a bit of statistical expertise. But again, a good first base is if you do have your own density estimates or even your own relative abundance estimates, it's a good idea to, compare, am I getting the same sort of hotspots as, what we're seeing in the statewide deer survey? Or, am I in the same ballpark with that? So, yeah.
Great. All right. Well, there's a few other questions, which there's a question around tree cover on private land, which might be more technical detail that's in the report, but that might be one that you follow up on later, Justin. And there's a kind of related to the control question, baseline question is also whether it'd be possible to use some of the species detection, other species detections to kind of understand the potential impact of deer over time. So I think the answer to that's probably yes, but I'll let you answer that one out offline. So folks, we're almost out of time. So thank you again, Justin. Great presentation, really informative. And also to Lauren as well, both, as someone put in the chat, quite different approaches to looking at understanding the distribution of feral animals and also their impact, but both highly relevant to management decisions, and hopefully we can take some of this work forward and feed that into various pest management programs that are operating in Victoria. So thank you both for the fantastic presentations. So our next ARI seminar is on Monday, the 27th of May, in a couple of weeks. It's Reconciliation Week that week, so it's a particular theme around working together to understand the impacts of cultural burning. So I'm really looking forward to that one. Please make sure that you're signed up to our seminar mailing list so that you can receive the link to the recordings from this seminar and also register for the next seminar. The team's putting various links into the chat there, so thanks everyone for that, and thanks again for attending and the engaging questions. It's really been a great session that I've thoroughly enjoyed as well. So thanks to our presenters again and for everyone attending. Have a good week.